to be here today. I will present Dr. Earl Miller. He's a professor of neuroscience at the Picower Institute for Learning and Memory and the Department of Brain Cognitive Science at MIT. Dr. Miller uses experimental and theoretical approaches to study the neural basis of high-level cognitive function that underlie complex goal-directed behavior. He focused on the frontal lobe, the most elaborated region of the human brain that is linked to neuropsychiatric disorders. Earl is an expert in working memory. I don't have any enough words to describe how important is his work in this field. So today he will talk about something else about working memory. So thanks, Earl, for accepting our invitation, and please, you can start talking when you want.
Thank you very much for the kind invitation. I really appreciate the chance to speak to you. And yes, I will be talking today about working memory. We all know what working memory is, perhaps especially this group. Working memory is the ability to hold things in mind. It's the foundational mechanism that allows higher level cognition, because with working memory, an important thing to remember about working memory is that it's not just a short-term memory buffer, it's under volitional control. And that's the most important thing about working memory, because working memory allows us to take in information and wait to make a response, maybe take in more information, decide to respond later, hold off decisions. It allows us, working memory is the fundamental mechanism that allows us to not just react to the environment, but to wait and act when we want to act on the environment. So it's very critical to high-level cognition. So the question today is, how do we hold thoughts in working memory? Now, the classic model for the past 50 years or so has been persistent spiking of neurons. The idea here is that when you're holding something in working memory, whether it's a visual image or it's a set of numbers you have to type into a keypad, you're doing it by having neurons in your cortex that are constantly firing, keeping those memories alive, keeping them in working memory. And that's illustrated here from a classic figure from Pat Goldman-Rakesh, one of the pioneers of the neural basis of working memory. And what it shows here is a typical neuron that has been used to support this persistent spiking hypothesis. This is one neuron. It's averaged across multiple behavioral trials. The neuron spiking rate is on the y-axis. Time is on the x-axis. Here's where you present the monkey, in this case, it's monkeys, with a visual cue that they have to hold in working memory. Then there's a memory delay, and then the monkey makes its response. So what typically happens is you present the animal with a cue, you get an elevated spiking, reflecting that cue. Once the cue goes away, spiking drops down a little bit, but it's maintained at a rate above baseline rate. And this presumably is keeping the ensemble, the memory, the information about that cue online and available in working memory over this memory delay until the animal makes its response, then activity drops back down to baseline. Now, this has been the dominant model for the past 50 years or so, and it's mainly based on averaging across, in this case, one neuron across multiple trials, or multiple neurons across multiple trials. However, over the past 20, 25 years or so, there's been great advances in multiple electrode technology that allows us to record not from one neuron at a time, but multiple neurons at a time, up to hundreds of neurons at a time. And this allows us to have a more detailed examination of what is happening in real time when animals are holding information in working memory. And what's especially important here is what's happening in real time on single trials. Neurophysiologists average across multiple trials because they want to increase the signal-noise ratio. The idea is that averaging brings out the signal and all the noise cancels out. But what if some of that noise is actually signal that we're just averaging out? And so Our lab and a number of labs have been looking at neural activity in real time on single trials. And what this has revealed is that in actuality, most neurons in prefrontal cortex and other higher cortical areas, prefrontal cortex being the frontal lobes, it's the part of the brain that's most important for most central working memory. Not the only part of the brain, but it's a central part of the brain. And what this has revealed is that most neurons in places like the prefrontal cortex, parietal cortex, higher level cortical areas that contribute to working memory, most of them don't show real robust, persistent, constant firing over this memory delay. Instead, they show very sparse and bursty firing. And I'll show you an example of that in a moment. But this observation that if you look really carefully at a finer grain detail, that most neurons do show this kind of sparseness, not persistent activity. It looks persistent if you average a bunch of bursty neurons together or average them together across multiple trials. But this observation that it is on single trials in real time that this activity is sparse and bursty has led to several updates to the classic model. One update came directly from Pat Goldman-Rakish, the pioneer of working memory, of the real basis of working memory, who in one of her last papers showed that it's not just spiking that seems to carry working memories, it's spiking plus spiking-induced temporary changes in synaptic weights. And she showed this in biology of the prefrontal cortex. When neurons in the prefrontal cortex spike, they spike, and then for about 750 milliseconds or a second later, there's a temporary potentiation of the synaptic weights between connected neurons. And that, Goldman-Rakish and Wong hypothesized, may help carry working memories in between these episodes of spiking. There's also the activity silent model of Mark Stokes, who makes the same sort of argument, and a recent model called the dynamic attractor model of Michael Lundquist. And I'll tell you about that dynamic attractor model in a moment. But all these updates to the classic model say the same thing. They say that spiking is not doing all the work. Spiking is important. Elevated levels of spiking during this memory delay is indeed important for holding information in working memory, but the spiking is helped along by temporary changes in synaptic weights, temporary impressions that spiking leaves in the networks. And this makes sense because spikes cost energy. Every time a spike occurs, it's costing a lot of energy. So rather than have constant spiking carrying the working memories, there's occasional spiking, sparse spiking, and in between the sparse spiking, the changes in synaptic weights are help carrying those working memories. And that saves energy because, again, spikes cost energy. But it also makes working memories more robust, less easy to disrupt than if they were held by persistent activity alone. Persistent activity in working memory is modeled by something called attractor dynamics. And attractor dynamics simply model this persistent state of activity where the neurons are constantly firing. And one thing we've learned from all these models is that attractor dynamics, persistent spiking, is not a very robust way, not a very good way to hold information in working memory. Because if a distractor comes along or a new input that comes along, you want to add something to working memory, the new input disrupts the attractor dynamics, disrupts the persistent activity, and changes the memories. So models of working memory based on persistent activity are very easy to disrupt. And these activity silent dynamics are more robust because the synaptic weight changes are more resistant to interference. So here's what we did to test this model. I'll tell you about the dynamic attractor model of Michael Lundquist. Michael came to my lab to test this model. And the way we tested this model is we studied a task in which monkeys had to hold two visual items in working memory simultaneously, while we recorded from multiple electrodes in the lateral prefrontal cortex of the monkey, shown here. Here's the prefrontal cortex. Lateral part of the prefrontal cortex would be right here on my head. So this is anterior, that's posterior, that's dorsal, that's ventral. I assume you can all see my cursor. Yes? If not, please let me know.
Yes.
Great. And here's the task. The task is the monkey was shown in the encoding phase two samples, two colored squares at two different locations. Then there was a memory delay, and they were tested for the memory of those two samples by a test array, and the monkey had to make a same and different judgment. Were these two squares the same as the ones they saw earlier? Now, this design looks complex, but the important thing to remember is all that was required of the monkey was to hold two visual items, two colored squares in the location of working memory. That's it. Just hold these two items simultaneously in working memory. And here's what we found. This shows, now not showing spiking activity here, what I'm showing is power from the local field potentials. Local field potentials are the mass average of millions of neurons spiking together in a coordinated fashion. We looked at the spiking activity too, but what we found is illustrated best by looking at the local field potentials. So here's frequency on the y-axis. This is frequency of the local field potential, anywhere from 0 hertz up to 120 hertz. The power of the local field potential is shown in color. So red is more power and blue is less power. Time is on the x-axis. S1 and S2 is when the two squares are shown to the monkey. Here we're showing the monkey the first square. There's a short pause. The second square, the monkey has to hold these two squares in working memory over this memory delay and then make a response out here. And what we found is illustrated in this slide. We found, like other studies, that there was spiking activity that conveyed information about the two pictures the monkey was holding in working memory, the two items in working memory. But that spiking activity that carried the working memory was associated with bursts of gamma from the local field potentials. So whenever the spiking occurred that conveyed information about the two items, S1 and S2, there was also co-occurring these bursts of gamma. And you can see that here, these gamma bursts were, and the spiking was most evident when the monkey was encoding the picture in working memory during the time in which we showed the picture to the animals, and then periodically over the memory delay. We saw these sparse bursts of gamma over the memory delay, and those bursts of gamma were associated with the spiking that was carrying information about working memory. So the first observation is that the spikes carrying the working memories were associated with gamma bursting in the prefrontal cortex. Now, another thing you may notice that here's the spiking up here on the top of the slide in the gamma range from 40 hertz and above. There was also bursts of a lower frequency in the beta range from about 30 hertz to about 20 hertz. And that's what's shown here. Here's two examples of beta bursting in this lower range. And what we found is that the beta bursting was also there, but it was anti-correlated with gamma. So you see the gamma and the spiking that carries the working memories, that encodes the information in working memory and carries the working memory, is associated with these bursts of gamma. And there were bursts of beta, but the beta tend to occur whenever gamma wasn't occurring. So here we see two bursts of beta here, and they're occurring at this interval of time where there's no gamma. Okay, so beta and gamma are anti-correlated. And this is illustrated here, where now we're looking at not the individual trial, but the average across multiple trials. We're looking at the burst rate. How often do we see these gamma bursts shown in the orange color versus the beta burst shown in the brown color? And you can see they're completely anti-correlated with one another. Gamma bursting goes up when the monkey is viewing the two sample objects here during S1 and S2. The monkey is seeing S1 and S2, and there's a burst of gamma. The monkey sees S2, there's a burst of gamma. And whenever gamma bursting is going up, beta bursting goes down. And over the memory delay, what you typically see in the prefrontal cortex in spiking is you see there's a relatively low level of spiking early in the delay that builds up over the course of the delay as the animal gets ready to use the working memories. And you see that reflected here in gamma. The gamma bursting associated with the spiking starts out at a relatively low level, then builds up over the memory delay, reaching an apex near the end of the delay when the monkey has to then use the working memories. And we see a corresponding drop in beta bursting. So beta and gamma are like mirror images of one another. Whenever gamma goes up, that's the gamma that's carrying the spiking, that's carrying the working memories. Whenever gamma goes up, beta goes down. Whenever beta goes up, gamma goes down. So I've said repeatedly that gamma is carrying the spiking that's carrying the working memories. If gamma and spiking is associated with the contents of working memory, what does beta do? Well, the answer is that a number of studies in our laboratory have shown that beta rhythms, these lower frequency beta rhythms from about 12 to 30 hertz, carries top-down information, the animal's knowledge about how to perform the task. And this is first shown by Tim Bushman, who's now a professor at Princeton. He was a graduate student and a postdoc in my lab. He had the animal switch back and forth between two different rules. The animal was looking at the stimuli, had to make judgments about either the color of the stimulus or the shape of the stimulus. And we cued the animal on this trial, pay attention to color, ignore shape. Then on another trial, pay attention to shape and ignore color. So the monkey is following these two rules, color or shape. And what Tim found is that this is recordings from the lateral prefrontal cortex. Here's the arcuate sulcus. There's the principal sulcus. Posterior is here, anterior is here, dorsal, ventral. All these little circles are a different recording site where we had a single electrode. We had an array of electrodes. And what we're looking at here is, again, local field potentials, but we're looking at which recording sites showed an increase in synchrony whenever the animal was following the color rule versus the shape, or what we'll call the orientation rule. It wasn't shape, it was the orientation of the item, but essentially the same thing. And what we found is that when we look at these patterns of LFP synchrony across the prefrontal cortex, we see different patterns of synchrony for one rule versus the other rule. Okay, so different shifting patterns of beta synchrony. And we looked across all frequencies to look for this effect, and we only saw it at the beta range, 12 to 30 hertz. But there's different patterns of beta depending on which rule the monkey was following, as if these patterns of synchrony are forming the ensembles needed to solve this task, needed for the animal to follow these rules. Okay, is there a question? No. Okay, so once we saw this effect for these rules, we then applied it to other forms of top-down knowledge. We saw the same thing for spatial categories, taught the monkeys the categories up versus down, and same things, different patterns of beta coherence in the prefrontal cortex and the parietal cortex, and between the prefrontal and parietal cortex, when the monkey was making a judgment about one category up versus the other category down. We saw the same thing for shape categories. We had a study where we taught animals to recognize the categories cat versus dogs, and the same thing, different patterns of beta coherence across the prefrontal cortex whenever the monkey was judging cat versus judging dog. And finally, other examples of shape categories, this was formed by dot patterns. The take-home message from this slide is that whenever we teach the monkey some rules, some knowledge that it needs to solve the task, not simply, the animal's it's not a matter of the animal viewing a picture. Whenever the animal is using some acquired knowledge, some top-down knowledge to solve the task, we see this top-down knowledge reflected in patterns of coherence in the beta range, in the prefrontal cortex, and in other cortical areas. So what this suggests to us is that we have this push-pull relationship between beta and gamma. Gamma is carrying the contents of working memory. It's involved in encoding information in working memory and maintaining information in working memory, and it's anti-correlated with beta. So the idea here is that top-down information is carried by beta, and they play an inhibitory role that controls the gamma that encodes information in working memory and maintains information in working memory. So it's almost like a beta is this control signal that turns on and off the faucet of working memory storage. Okay? So beta has this inhibitory relationship to gamma. If you want to encode information in working memory, you drop down beta. That allows gamma to be expressed. That encodes information in working memory. you want to maintain information in working memory, you keep beta at a low level. And if you want to drive up the contents of working memory, like we see over this working memory delay, so the animal can get ready, when the animal's getting ready to use the working memories, we drive down beta, it allows more expression of gamma. Okay, so it's a beta acts as a control signal, inhibitory control signal that turns on and off working memory storage. That's the basic idea. And please, if you have any questions, not being clear about anything, feel free to interrupt me. Okay, so once we have this observation, we want to explore this further. This anti-correlation between beta and gamma suggested, we showed the anti-correlation between gamma and beta. We showed that gamma carries working memory, beta carries top-down information, but do they really work like this push-pull control signal that actually controls working memory? So we did this in a more recent study where we did a very complicated working memory task. I'm not going to walk you through, in the limited time we have here, I'm not going to walk you through this particular study. I'm going to highlight the results, and I invite you to read the paper if you want more details. But what we found is that evidence that gamma and beta and gamma work together to gate information in working memory. So first of all, we showed that beta gates information into working memory. Whenever the animal wants to encode information in working memory, beta drops. It allows gamma and spiking to increase. And you see that right here. Here's the animal has to encode the stimulus in working memory. Beta drops. It allows gamma and spiking to start encoding the information in working memory. So that can be seen here. Well, we also showed in this study in 2018 in Nature Communications that beta is also involved in clearing out working memory. If at some point in the trial, the monkey is cued to know that the working memory is no longer relevant, the monkey can just forget the working memory. What happens is there's a big increase in beta that drives gamma down, spiking stops, and working memory is cleared out. Okay? Another thing in this particular study we did is we had a trick where we had the animal switch the contents of working memory. First, the monkey was holding one stimulus of working memory. Then we gave the animal a cue, forget about that stimulus and code a second stimulus of working memory. And when that happened, there was this rapid cycling of beta gamma. That first beta went up and dropped down the spiking in gamma to the first object. That cleared the first object out of working memory. Then beta dropped, gamma increased, and the second item was then encoded in working memory. So this study in 2018 shows that you do see this push-pull, this dance between beta and gamma that is very suggestive of a control signal that controls the encoding and maintenance of information in working memory. Right? And importantly, what neurophysiologists have used for many years to see whether the spiking effects or any effects they're seeing are actually related to the task at hand, in this case, related to the working memory task, we look at error trials. If the animal makes an error, can we see the error reflected in whatever activity we're looking at? And that was true for this beta-gamma balance. In fact, we looked at individual trials and tried to predict what mistakes the monkey would make, what decision the monkey was making. We found that the balance between beta and gamma was a better predictor of the monkey's decisions and whatever errors the monkey would make than spiking activity alone. So beta and gamma was highly predictive of what decisions the monkey would make, even when the decision was wrong. Okay, so we wanted to look more closely at the dynamics between beta and gamma, and we did so by doing so-called multilaminar recordings across using multiple contact electrodes along the layers of cortex. And this is relevant because the superficial layers of cortex are the feed-forward layers that carry sensory information from the back of the brain to the front of the brain. Whereas the deep layers of cortex are the feedback layers that carry top-down information from the front of the brain to the back of the brain. And to make a long story short, what we found is that these dynamics were reflected in different layers of cortex. So the spiking activity that was carrying the working memories was mainly in the superficial layers of cortex. Here's the different cortical layers. The dotted line shows the middle layer, the input layer, layer 4. The color in this case shows spiking activity. Here's a working memory delay where the monkey's seeing a cue here. This is the memory delay. And we see there's spiking that's carrying the working memory the monkey's holding, in the stimulus the monkey's holding a working memory, but that's mainly in the superficial layers of cortex, not the deep layers of cortex. And that makes sense because the monkey's holding a sensory stimulus, a working memory, so that should come, since it's feed forward sensory information, it should be in the superficial layers, and it is. And then we looked across the different layers again. Here's the different layers again, with 0 being the dotted line being the middle layer. We saw more gamma in the superficial layers and more beta in the deep layers. And this makes sense because, again, gamma is associated with the sensory information the monkey's holding a working memory. Superficial layers, the feed forward layers, since gamma is associated with holding sensory information and working memory, it should be stronger in the superficial layers, and it is. And beta is carrying top-down information, we hypothesize. And because the deep layers are the feedback layers of cortex that carry top-down information, it makes sense that beta should be stronger in the deep layers of cortex, because it's carrying top-down information feeding back from the front of the brain to the back of the brain. And the same beta gamma anti-correlation I showed you earlier, we saw taking place across the superficial and deep layers of cortex. To save time, I won't walk you through this slide, but what this figure shows is that whenever beta in the deep layers of cortex was strong, gamma in the superficial layers is weak. And whenever gamma in the superficial layers was strong, beta in the deep layers was weak. So again, this push-pull relationship between beta and gamma, but happening between the two cortical layers. So what this suggests is the following. Top-down information is carried by deep layer beta from the frontal lobe back to sensory cortex. Deep layer beta regulates weaker superficial layer beta. We see beta in superficial layers too, but it's a weak compared to deep layers. So we think that the superficial layer beta is entrained by deep layer beta, and the superficial layer beta in turn inhibits, gates, or controls the gamma spiking carrying working memory stored, thereby turning on and off working memory. Okay? So that's the basic model. Now we have this top-down beta information acting as a control signal that gates the expression of gamma and spiking that encodes information in working memory and maintains information in working memory. We decided to carry this further in a very recent experiment just published by looking at a curious thing about visual working memory. Now, you probably all know that vision is split between the right and left brain. The two halves of both retinas go to different sides of the brain. The right half of each retina goes to the left brain, and the left half of each retina goes to the right brain. Bottom line being that your right brain sees one side of vision, one side of gaze, and your left brain sees the other side of gaze. Now, it's long been thought that this split between vision is well known for visual processing back in posterior cortex, and certainly in area V1 and primary sensory cortex. And there's been a sort of a tacit assumption in the field that somehow this split left and right gets united at the level of higher level cognition and prefrontal cortex. But we found out back in 2011, again, work done by Tim Bushman, Simon Kornblith, and Demetrius Panissis in the lab, they showed that the split between right and left is even true at the level of the prefrontal cortex and in working memory. In fact, your right and left sides of vision have separate working memory capacities, as if the monkeys, and we've replicated this in humans, as if monkeys and humans, as if we are split brain patients, two separate brains, one looking at the left side of vision, one looking at the right side of vision. So that's weird. We have right and left is split between the right brain and the left brain. But if that's the case, how do you actually unite the left and right sides of vision? When I'm looking around, when I'm playing Frisbee, when I'm just operating in my daily life, I don't notice a split between right and left. So how does the brain do it? So we trained monkeys on this paradigm where we had them encode information in one side of vision then did a little behavioral trick that encouraged them to switch the working memory from one side of the brain to the other side of the brain. So here's what we did. We had the animal fixate its gaze on one side of the computer screen, and we presented a stimulus the monkey had to hold a working memory to the one side of that gaze. In this case, this monkey is looking right here. We're presenting a banana to be held in working memory on the right side of vision. That means it's going to the left brain and not the right brain. Then on one half of the trials, we played a little trick on the monkey. On half of the trials, when the monkey was not, the banana is already gone, the monkey's just holding the banana in working memory. And while the monkey was holding the banana in working memory, on half of the trials, we had the animal move its gaze to the other side of the computer screen. Now, if the banana was still on the computer screen, when the monkey moved its eye to the other side of the screen, that should switch the vision from one side of the brain to the other. But in this case, the monkey is not looking at the banana, the monkey is just thinking about the banana. What happens there? Does this move the stimulus from the one side of the brain to the other? Well, the answer is that it does. So here's the two possibilities here. What we have here is neural activity, the contralateral stimuli are stronger than the ipsilateral stimuli. That's true all over visual cortex. It's also true for motor cortex. Your left side of your brain controls the right side of your body, so contralateral stuff is a stronger representation. One possibility is shown here is the dark lines, the dark green line, the dark brown line, are what happens on the trials in which there is no saccade and the memory just stays in the original hemisphere in which it was encoded. So in this case, that'd be so you should see a stronger response to contralateral stimuli, a weak response to ipsilateral stimuli. And what the lighter colored line shows is what happens when we have the animal make the saccade and the memory switches from the contralateral hemisphere to the ipsilateral hemisphere, or in the case of light brown, from the ipsilateral hemisphere to the contralateral hemisphere. Now, one possibility, this little trick does nothing. The memory just stays where it is. And we expect to just see no change whatsoever. But the other possibility is that the memory switches. So in this case, if the memory switches from contralateral to ipsilateral, we should see this flip where the stimulus started out with a strong representation because it was on the contralateral side. Then the monkey makes its saccade, shown here in this line right here, and that should flip the memory to the ipsilateral side, and then its response should weaken. Correspondingly, if the stimulus started on the ipsilateral side, shown here by this light brown line, and then the saccade moves the memory to the contralateral side, it should suddenly get stronger after the saccade because the memory is now switched to the contralateral hemisphere. And what we found is this: the memory didn't switch to the other hemisphere. So that's shown here. Here's the trials in which here's the beginning of the delay. You see the stronger neural responses to the contralateral stimuli, to the versus ipsilateral stimuli. This is before the saccade, in the middle of the delay. And this is the trials in which the saccade occurred. And first there's a burst of activity of the saccade, then the memory switches from the contralateral hemisphere to the ipsilateral hemisphere. And indeed, we see this dropping down of information, of neural activity. Once the saccade is made, the memory switches, neural activity drives down to the same level that you'd expect if the stimulus was on the ipsilateral side to begin with and never moved. And correspondingly, if a stimulus starts out on the ipsilateral side, then the saccade occurs. There's an increase in activity to that stimulus to the same level that you'd expect if the stimulus just started out in the contralateral hemisphere. So you see the switching operation. Contralateral stimuli after the saccade switch to the ipsilateral hemisphere, and ipsilateral stimuli after the saccade switch to the contralateral hemisphere. So the memory traces, once the saccade is made, the memory traces disappear from the ascending original hemisphere and then reappear in the opposite hemisphere, the receiver hemisphere. And interestingly, the same dynamics we saw for beta gamma dynamics we saw for working memory gating are also true for transfer of working memories between the hemispheres. Here's around the time the memory is being transferred, and around the time of that memory transfer, there's a decrease in beta power and an increase in gamma and theta power. Gamma and theta tend to co-track in cortex. Gamma and theta they work together. So we see this decrease in beta and increase in gamma during the working memory transfer, just like we saw for working memory maintenance. If you want the memories to transfer from one hemisphere to the other, you drop beta down, and that allows the gamma to then transfer and be expressed in the other side of the brain. And a Granger's causality analysis revealed that the directionality of the synchrony was consistent with memory trace transfer. So here's a member of the Granger causality here on the y-axis. This is for the sender to receiver hemisphere. The memories are going in the direction that we'd expect versus the opposite, receiver to sender. The memories are going the opposite direction, not the direction we'd expect. And here's the frequency of the LFP synchrony on the x-axis. And as you can see, the Granger's causality measure is stronger in the sender to receiver direction than the receiver to sender direction. It's stronger in the direction as if these synchrony is playing a role in pushing the memories from one side of the brain to the other. Okay? And the last thing I'll tell you about is the most surprising thing that we found that when these memories transfer from one side of the brain to the other, it actually changes the ensemble, changes the pattern of activity that represents the memory. So what we did is we trained a classifier on the pattern of activity across an array of electrodes in the prefrontal cortex. And we trained the classifier on the trials in which the stimulus started out in the contralateral hemisphere and then just stayed in the contralateral hemisphere. It didn't go anywhere, just stayed in the contralateral hemisphere. Then once we trained the classifier on those trials, we then applied it to working memories that were in the contralateral hemisphere, but were transferred over from the ipsilateral hemisphere. Okay, so we're training the classifier on memories that started on one side of the brain and stayed there. And then we're testing that classifier on memories that were received from the other hemisphere. And the hypothesis is that you get the same pattern of activity. You simply copy the working memory ensemble from one side of the brain to the other, and it doesn't change. You're simply copying it like you're taking a picture of it and just pushing it over to the other side of the brain. But the null hypothesis is that there is no transfer, that the working memory actually changes when it's transferred. And that's exactly what we found. So here's the trials in which we trained the classifier on the working memories that stayed in the contralateral hemisphere, and then tested them on models on working memories that started in the ipsilateral hemisphere, but then transferred to the contralateral hemisphere. And there was virtually no transfer at all. And what this says to us is that the classifier trained on visually invoked activity in the contralateral hemisphere cannot be used to decode the exact same working memory, same object, same location, same everything, but it can't be used to decode that working memory if that working memory was transferred from the other hemisphere. So it seems as if when the working memory is transferred from one hemisphere to another, its pattern of activity, its ensemble, completely changes to a new format during this transfer. So I see I'm running short on time, so I'll skip the last experiment just to say that we found that the same beta gamma dynamics not only has a role in working memory control, it also plays a role in something called predictive coding. And I encourage you to look up this paper in PNAS published last year, showing the same beta gamma dynamics play a role in how the brain regulates processing of sensory information using predictive coding. But to sum up, since I'm out of time, I'll say that we believe that these studies show that alpha beta is like a control signal that feeds top-down information in the deep layers of cortex from the front of the brain to the back of the brain. These top-down alpha beta signals, and by the way, I'm using alpha beta here because when you look at the back of the beta is primarily in the frontal cortex. When you move to back most posteriorly, there's a down regulation of frequency, so the beta in the frontal cortex becomes alpha in the visual cortex, but it's essentially the same signal. So these alpha beta control signals carrying top-down information regulate the expression of gamma, that carries the spiking, that carries bottom-up information, the contents of working memory or the contents of perception. And we believe this is a modal cortical circuit that plays a role in top-down control of basically everything. Working memory, attention, predictive coding, and even the transfer of working memories between the hemispheres. We have looked now, our original studies were in the frontal cortex and focused on working memory. We have looked for these dynamics all over cortex, and we're finding it everywhere we look. Every time there's a bottom-up sensory information, more gamma. Every time you need to regulate that sensory information, there's an increase in beta or alpha, depending on where you are. And that beta and alpha plays this inhibitory, permissive gating role on the gamma, beta that's carrying bottom-up information. So we think that this general circuit here is a modal cortical circuit that allows top-down information in your brain to control the processing of bottom-up information, allows you to control what you perceive, what you think about, and what you hold in memory. And with that, I thank you for your attention. And I thank all the people in my lab who actually do all the hard work conducting these studies. Thank you very much.
Thank you, Earl, for your interesting and excellent presentation. And we have a couple of questions. The first one is, what is the neural substrate of deep layers operating at beta and superficial layers operating at gamma?
Sorry, could you one more time, please?
What could be the neural substrate of deep layers operating at beta, and superficial layers operating at gamma?
That's a great question. And so where beta and gamma comes from is a great question. And that work is best addressed by, so far what we have is modeling work. And I turned you to the modeling work of my collaborator, Nancy Capel. And basically what you get is you get these recurrent connections between neurons, and then clusters that represent the same stimulus. They're strongly recurrently connected, and they inhibit other clusters, other ensembles that represent another stimulus. And whether you get beta or gamma depends on the EI balance, the balancing excitatory and inhibitory influences in those circuits. And what's interesting, if you couple together these different ensembles, so you have them oscillating different frequencies, and you couple them together, you get this interesting phenomenon called traveling waves. These beta and gamma waves don't just stay in one place in cortex like a jump rope. They actually move across cortex when you connect these modules together. And so in some recent study we're just doing right now, we're finding that these oscillatory dynamics in cortex form traveling waves that move across the cortex in a task-dependent way.
Okay. So another question that it occurs to me is, how do you, if I understand correct, the increasing the beta oscillations is a kind of eraser of the memories already stored in the working memory, in the working memory. Right. So my question is, what is the effect of this surge in beta oscillations into these layers presenting the beta oscillations? So kind of what is the mechanism that is inducing these oscillations?
Well, in the original Lundquist model, which we based our studies on, beta and gamma aren't actually separate circuits. They're different states of the same circuit. So a circuit is in beta when it's in a low energy state, when there's no bottom-up inputs. And then when you have a bottom-up input, the increased energy knocks the network up in the gamma range, and that may explain this difference between this push-pull between beta and gamma, because they're two different states of the same network. So what you have is you have the spiking carrying bottom-up information that's going to tend to drive the superficial layers of cortex up into the gamma range. Then you have the beta carrying the top-down information that's has less energy. It's going to be in the beta range. As for the exact circuitry that allows beta to inhibit gamma, that is still a subject of investigation. We don't know the exact circuit, but it could be a matter of the beta dynamics switching this EI balance to turn on and off the expression of gamma and spiking. And one thing I'll mention is that for most of the talks, I didn't get to the predictive coding part, I've treated this beta signal, alpha beta signals, if it's just a faucet turning on and off working memory. Our predictive coding study shows that it's not just as it's much more precise than that. It actually targets the representation of specific stimuli. You can actually target, turn on and off the representation of individual stimuli using these beta gamma dynamics. So it's not a general faucet. It can actually be used to manipulate individual stimuli.
Thank you. And we have another question from the public. Jose, please, you can ask.
First of all, thank you, Earl, for your talk. Very good, too good. I have just one idea. And you started giving the example of visual cortex. Yep. And in visual cortex, you have this very classic and very well-known retinotopic representation. You show in your last part of the talk that may not be the case for working memory, at least in the prefrontal cortex, because you don't transfer in the experiments where you train the classifier, you don't get the same activity in one side and in the other side.
That's right.
Which would be expected for a nemotopic, if you can say, cortex, if the representation is exactly the same at one side of right. or the other, like more or less visual cortex regarding retina.
Right.
Two questions. It may seem the easy question is where do you think if we at some point of the building of memories we need this spatial coherence between the two sides or not? Maybe not in the prefrontal cortex, but at other levels. And two, it may not be the case that activity, giving beta and gamma dancing, if you can say, is more important than the cells, the individual cells, at least in prefrontal cortex, not the case for visual cortex, V1.
Mm-hmm. Well, actually, could you unpack the last part of that question? What do you mean by the spikes, beta and gamma not being as important as spiking?
Meaning that it doesn't matter who is spiking, but the rhythm is which is in that particular cell engaged.
Well, I will say that, when we look for information about sensory information, for example, we don't find much information per se in gamma, in rhythms, in spike. Gamma seems to be just co-expressed with spiking, and we only can really decode information about the stimuli when we look at spiking. Okay. But alpha beta is a different story. Alpha beta, you can actually get top-down information. You can decode top-down information from the patterns of alpha beta. So there seems to be some difference there between alpha and beta can carry top-down information, but when it comes to sensory information, that's largely tied to spiking, but the spiking is associated with gamma. It helps traffic spiking. As for the first part of your question, it's true that we've known for a long time that prefrontal cortex is not retinotopic. But what we found in our studies, people sort of assume that maybe when you get to the level of prefrontal cortex, you got these huge bilateral receptive fields of neurons. So the neurons then bridge the gap between left and right. But that assumption was based on some very early studies in anesthetized animals. And what we learned is that the anesthetized brain is very different from the awake brain. And when we did these studies looking at the representation in the prefrontal cortex in the awake brain, there's no retinotopy, but there is a strong, heavy contralateral bias, just like you see back in the visual cortex. I'm sorry, did that answer all your questions?
Yeah. What have you? I will make another question.
Okay.
Did you know what happened with monkeys or animals who have the corpus callosum, I don't know, slice or cut? Because where do you, anatomically, I'm thinking, where this transfer goes by?
Yeah, that is a great question. And we just now planning a series of experiments where we're going to record from all over cortex and look to see where this transfer is taking place. One possibility is it takes place in the prefrontal cortex. Then once a memory goes from one side of the brain to the other, and it gets fed back to the rest of the cortex. Another possibility is the transfer takes place at every level of cortex because there's connections to the corpus callosum at every level of the cortex. So the first thing we're going to do is we're going to do a neurophysiological survey of every level of cortex, prefrontal, parietal, visual cortex, different levels of visual cortex, and see where we see the evidence of this neurophysiological evidence for this transfer between the hemispheres. And then we'll have a better idea of what sort of manipulation experiments we can do to nail down where the transfer is taking place. I would hypothesize, I would guess that we're not going to see this, probably not an exclusive function of the prefrontal cortex. We'll probably see some active transfer even back in visual cortex. And in one of these studies, we're planning on not just using working memory, but having the animal trace objects as they move back and forth between the right and left visual field. And we want to see if that gets handed off between the right and left visual field, not just in the PFC, but at different levels of cortex.
Sounds very, very cool experiments and very cool hypothesis. Yeah. Good luck with that.
Thank you.
Okay, we have another question from the public, and is from Claudio Villalobos. He says, are beta and gamma oscillations independent or a correlated phenomenon? Would be reducing or deleting gamma would have any effect on beta oscillation?
Well, all over cortex, you see that beta or alpha, it's beta in frontal cortex, but alpha in visual cortex. And we've done a survey across cortex, and what you see is as you move from the front of the brain to the back of the brain, all the frequencies gradually drop down a little bit. So beta and alpha are pretty much the same thing, just in parts of the brain. All over cortex, you see beta, alpha, and gamma are anti-correlated. And the classic example is in motor cortex. You have the animal, or human for that matter, you instruct them to make a motor response, but say, don't make it yet, hold on, wait for a go signal. You'll see lots of betas are suppressing the control signal, the motor response. And then as soon as they're giving the go signal, beta drops, gamma goes up. And there's also studies in visual attention. Whenever the animal, say the animal attend to a stimulus, lots of gamma, have the animal attend somewhere else, what goes on is alpha goes up for the to the place where you're not paying attention to. So these dynamics between alpha, beta, and gamma are anti-correlated all over cortex. So we think they're correlated in the sense that they're anti-correlated. They seem to be, if they're not inhibiting one another, they're at least different parts of the same, different states of the same network. So they're correlated in the sense that they're anti-correlated. You don't see many examples of, or any examples I know of, where higher frequency gamma and lower frequency alpha beta are actually coexistent. You do see cross-frequency coupling where different frequencies will work together, but not for alpha, beta, and gamma. They tend to be really anti-correlated. You see coupling between like very low theta and gamma, so across a wider range, but not alpha, beta, and gamma per se. They seem to be, everywhere you look, they seem to have this anti-correlated push-pull kind of relationship.
Okay, and the last question is that, there is any mechanism, or any task where the whole prefrontal cortex is engaged in this gamma or beta oscillations at the same time in both sides of the brain, not just one or two?
Oh, now that is a great question. That's another study we're doing. So I presented as there's separate working memory sourcing in the right and left sides of vision. And actually, my colleague Ed Vogel has studied 1,000 people now, and we find the same split in the human brain. That it really is as if we're all a bunch of split brain patients when it comes to visual working memory. Okay. Do we see any place where it's happening at the same time? Well, one example is what happens if I holding a stimulus of working memory that I look at directly at the center of vision, which is the way working memory has been studied for 50 years, or at least non-spatial working memory. You just look directly at a stimulus. What should happen there is the working memory should go to both hemispheres. So what happens? Are they coordinated between the hemispheres? Are they anti-correlated to the hemispheres? Are they independent? Nobody knows, because amazingly, we look back over the history of monkey neurophysiology, and no one has recorded bilaterally in the cortex. They've done what we all, probably what you guys have always done, what we always done. We record from one side of the brain and save the other side for a future experiment. So no one knows the answer to these questions because no one's ever recorded bilaterally in cortex. So this completes some studies where we did the classic working memory study, just hold a picture that was presented at the fovea while we recorded bilaterally. So I literally should have an answer to you in about a month or so.
Okay, thank you. Thank you for your really excellent talk. And okay, in honor of time, because we are late, we want to thank you for your willingness to present here. So thank you.
Thank you.
Thank you, sir.
Thank you for the invitation. I really appreciate it.